Okay then, this is the next revision video that I was asked to do, which was gas exchange in fish. So it follows directly on from gas exchange in insects. Okay, so just before we start, like I did with the insects, it's really important to remember the Ficks Law. Okay, because all of these gas exchange surfaces depend on the principle stated in Ficks Law, which is that the rate of diffusion is proportional to surface area multiplied by the concentration gradient or the concentration difference, surface area, divided by the distance of diffusion. Distance of diffusion, sometimes referred to as diffusion pathway. I prefer distance because that's actually a measurable value. Okay, so when we look at gas exchange in fish, we need to consider how to increase that rate of diffusion. So we need a larger surface area, maintain a concentration gradient, and a short distance of diffusion. Okay, so we're going to have a look at the structure and explain how those structures and how those adaptations enable a fast rate of diffusion. Okay, then we will compare the countercurrent and parallel flow and explain why countercurrent in bony fish is a more efficient method for exchanging gas, for exchanging oxygen and carbon dioxide. Okay, so let's get moving on. So it's necessary for fish to have a specialised exchange surface, and in fact this might relate to a number of different organisms. Firstly, they're large, so the distance of diffusion to reach those internal tissues is much too great. Secondly, they have a waterproof surface. Okay, so it's gas tight, and that idea that the distance is too great, that's because they've got a surface area to volume ratio that's too small to exchange across their surface alone. Okay, so there are two methods of ventilation. Movement of air in and then out, and then in and out, and repeated, is referred to as tidal. Tidal, like the sea coming in and moving out with the tide. So we see that in mammals. We describe that in insects, where air moves in to the gas exchange surface and then moves back out again, referred to as tidal. Another method is what's called unidirectional. So uni meaning one, so moving in one direction. So the fluid moves over the gas exchange surface in just a single direction, whereas here the fluid would move in and out. Okay, so mammals, and as we said, insects, they show this tidal ventilation. So air moves in, for example, into the lungs, into the alveoli, gas exchange takes place, and then air moves out again. In fish, they show a unidirectional ventilation, where water moves into the mouth, over the gills, and then out of the fish through a space called the operculum, which is a muscular tissue that we'll have a look at in a moment. And it always moves in one direction. Now the reason for that or the necessity for that, is that water has a much greater density than air. Therefore, it will require too much energy and too much movement brought about by muscles for it to be efficient if water were to move in and then back out again. So fish show this unidirectional ventilation, where the water moves in one direction because of its greater density. Okay, so the external anatomy of a fish then. Here's the mouth. Okay, so the fish, the water moves into the mouth. I'll explain in a little bit more detail shortly about how that happens, and then passes over the gills and then out through this slit here, which is called the operculum. So if I can do it in red, it might be easier to see. Okay, so there is the operculum. And the operculum can be opened or closed. It's muscular, and the fish is able to open or close the operculum. And we'll have a look at the stages that take place for that to happen shortly. Okay. Now this fish is actually a bony fish, or its proper name is a teleost. Don't need to know that, but it's interesting. Now bony fish, so these are the majority of fish that you probably think of. So if you think of cod or barracuda or I don't know, a goldfish even, are all referred to as bony fish. The reason they're called bony fish is because their skeleton is made out of calcified bone. Sharks and rays, guitarfish, etc. are cartilaginous. Okay, and they don't have an operculum. They don't have this muscular opening and closing. The cartilaginous fish have what are called gill slits, and those gill slits freely open. You might have seen that if you watch a shark swimming forward, you might see the those gill slits open, allowing the water to move through. Okay, we're going to focus on these bony teleost fish. Underneath the operculum, so there is the operculum, there's the muscular part. Underneath we can see these gills. Okay, and they're on a number of arches. Okay, so here I think we can see 3. The number of gill arches, or branchial arch, this bony bar that we can see in the notes, changes. Okay, depending on what the fish is, but the example that you've been given here, surface of the gills consists of 4 pairs of gill arches. Okey-doke. Right, now already, as there are 4 of them rather than just 1, we've already got an increased surface area. So there are 4 gill arches. Let's have a look at those gill arches in a bit more detail. Okay, so the gill arch is this larger structure. These are the gill rakers. So the gill rakers prevent any food from passing into the gill and causing a blockage. On top of the gill arch, you can see these lines. Let's see if I can draw that in a different colour. Okay, shown here. These black lines. These are the gill filaments. And we can see there are many gill filaments. Okay, so many gill filaments. probably guess. Again, increases the surface area. But again, that's not enough. Okay. On top of those gill filaments are, if we have a look at the notes here, are another layer or another level of structure called the lamellae. And there are many lamellae on each of the gill filaments. So many lamellae further increase the surface area. So if we go back to our original Ficks law, we've got a large surface area of the gas exchange surface due to the, okay, so we've got many, perhaps 4 gill arches, 4 pairs, so 4 on one side, 4 on the other side. On each of those gill arches are many gill filaments, and then on each of the gill filaments there are many lamellae, all of which increases that surface area. Okay. So, water continually flows over the gills. So it flows between the arches, over the filaments and over the lamellae. That's really important. Okay, so that water moving over the gills supports those gills and prevents them from collapsing on top of each other. Okay, so the reason a fish will suffocate out of water is because their gill filaments and their lamellae stick together. And when they stick together, that decreases the surface area, and therefore they're unable to do efficient gas exchange, or unable to diffuse oxygen from the air and move carbon dioxide from the blood into the air. And you can kind of see that in this diagram here, I think. Where is it? Okay, if we look really carefully, you can see that the filaments have all stuck together. Okay, if you suspend the gills in water, they will actually separate out, and we do this as a we may do this as a dissection at a later date. Okey-doke. So, water has oxygen dissolved in it. Okay, and that is really important. It is dissolved. I have asked people in the past whether they think fish are able to do this. In fact, every year I ask this. Can fish do this reaction? Can they split water into hydrogen and oxygen? And of course, they can't. Okay, but there's often 1 or 2 in the class that think that maybe they can, because they've not really thought about it. Often those people remain quiet because they realise at the time that it's not what's happening. Instead, you can consider the water that the fish is swimming in as a mixture, and in that mixture is some dissolved oxygen. But the oxygen content of that water is fairly low. Air contains about 21% oxygen at sea level. Water, perhaps 1% oxygen. And therefore, the gills have to be really efficient to extract the oxygen from the water. So, here are those adaptations again. They have, and this is the these are the responses you could use in an exam, okay, to describe the structures and to explain their adaptation. So, they have a very large surface area created by many gill filaments and many lamellae. Okay, and use that phrase, many gill filaments and many lamellae together. Just as an aside, you can see here this is a gill arch out of water, and you can see those filaments all stuck together, as I described earlier. Okay, here we have the filaments again. So the filaments are these longer ones, and on top of those, tiny lamellae. We'll have a look at those in a while. It's a large surface area. Now, the movement of the blood over the gill filament means that oxygenated blood is taken away from the gill and replaced by blood with a lower concentration of oxygen. This is this idea of maintaining a concentration gradient of oxygen. So the surface area and the concentration gradient have been increased. Can you see where we're going with this? Excellent. The capillary, the walls of the capillaries that are close to the lamellae is 1 cell thick. So it's 1 endothelial cell thick. The lamellae are 1 epithelial cell thick, and therefore they have a very small distance of diffusion. So the gases need to be exchanged over a single epithelial cell in the lamellae and a single endothelial cell to the capillary. Altogether, all of those features mean that there is an increased rate of diffusion. Okay, then you have this diagram in your notes. I've just mentioned that, so there's that the epithelial layer of the gill lamellae and the endothelial layer of the capillary, so just 2 cells separate the water from the blood. Large surface area, we've mentioned already, with the gill filaments, so many gill filaments on which there are many lamellae, which increase the surface area. Okay, we know that there is that ventilation, so the movement of the water continuously over the gills, which maintains the concentration gradient. So together, here are all of those features. As I mentioned, many filaments and lamellae, large surface area. And epithelial, lamellae, endothelial capillaries, so short distance of diffusion. Each lamellae has its own capillary network. Okay, that again increases the surface area, and there is a ventilation mechanism. We'll come back to that ventilation mechanism in a moment. Okay, here it is. Look carefully at the lamellae. So the lamellae is a tissue. In that tissue there are capillaries, and the arrows on the capillaries show the direction that the blood is flowing. So deoxygenated blood travels to the lamellae, it moves into the capillaries and moves across that capillary, but look at this, it's moving in the opposite direction to the blood. Sorry, opposite direction to the flow of the water. So as water moves past the lamellae, the blood is moving in the opposite direction. Move opposite direction to the flow of water. Now that is a really useful phrase, and we'll have a look at why in a second, but that is usually worth 1 mark. Okay, I'll just skip through that bit. Okay, now this idea that the blood is moving in the opposite direction to the water is referred to as countercurrent, or sometimes countercurrent flow. Before we move on to countercurrent, let's have a look at the other version. Parallel flow shows where the blood and the water flow in the same direction. Now, parallel flow is present in those cartilaginous fish, sharks, rays, guitarfish, etc. Okay, now if we have a look, the water, as it enters the or as it begins to pass over the lamellae, has a higher saturation of oxygen. It comes into contact with blood with a much lower saturation of oxygen, as we'd expect. So the oxygen diffuses from the water across the epithelial layer of across the epithelial cells of the lamellae, across the endothelial cells of the capillary, and into the blood. Okay, and it continues to do that as long as there is a concentration gradient. So here we can see we've got higher, oh actually I won't do it there, we've got a higher oxygen concentration in the water, lower oxygen concentration in the blood. But as the water and the blood are moving in the same direction, eventually, at some point along the length of the lamellae, equilibrium is reached. So it's only ever possible for 50% of the oxygen in the water to diffuse into the blood. Okay, so here are some useful little questions to think about. Does oxygen diffuse out of the water? I want you to think about this phrase: along the whole length of the lamellae. Remember, this is the lamellae, showing the lamellae. No, it doesn't. Okay, now that is a really useful phrase to say that in parallel flow, oxygen does not diffuse across the whole length or along the whole length of the lamellae. How much oxygen does diffuse? Well, 50%. Why can no more diffuse out of the water? Because equilibrium is reached. Okay, it does not maintain a concentration gradient. So the concentration gradient is not maintained. Okay, so that's parallel flow, present in sharks and rays, etc. Okay, this is another way of demonstrating that. Okay, on a graph, distance along the gill plate. Gill plate is the lamellae. As the water travels over the gill plate, initially oxygen diffuses across, but then it reaches equilibrium here, and there can be no more exchange of oxygen, and therefore the concentration or the amount of oxygen that can move from the water and into the blood is only ever 50%. Or is that most 50%? So that's parallel flow. This is countercurrent flow. And if you remember back to the diagram we looked at earlier, where is it? Okay, here we could see that the blood moved through the capillaries in the lamellae in the opposite direction to the movement of the water. Now, what we find there is water always comes into contact with blood with a lower concentration of oxygen. Okay, so this first mark in a question about countercurrent flow, you must say that the blood and the water flow in opposite directions. Use that phrase exactly. The second mark we can say is that the water has a higher concentration of oxygen than the blood across or along the whole length of the lamellae, and therefore oxygen will diffuse out of the water along the whole length of the lamellae. So this phrase, along the whole length of the lamellae, is really useful. In this instance, the maximum saturation reached is 90% in the blood. On average, it's about 80%. It depends on the species of fish and how sort of metabolically active they are. The important part, though, this second sentence here: oxygen diffuses out of the water along the whole length of the lamellae. Now we can see it graphically, so we can see that the water always has a saturation of oxygen greater than the blood. The water is moving in the opposite direction to the blood, and across the whole length of the lamellae, oxygen diffuses from the water and into the blood. Those three phrases should get you all of the marks in a countercurrent flow explanation. Okay, so number one, it's just must say that the blood and the water move in opposite directions. Number two, the water has a higher oxygen concentration. Number three, you might link 2 and 3 together. So oxygen diffuses, not across, diffuses from the water along the whole way, sorry about that, along the whole length of the lamellae. Great. Okay, if we compare that then, obviously the saturation of the oxygen in the blood with countercurrent flow can be much greater than it is with parallel flow. Parallel flow, a maximum of 50% saturation, or 50% of the oxygen from the water into the blood. With parallel flow, it can be much higher, usually around 80%, or on average. You might say here, equilibrium is not met in countercurrent flow. Okey-doke. You've got that diagram in your notes. You've got that diagram as well, and I think you might even have this one. So here are those 3 points again. Number one, blood and water flow in opposite direction. Number two, this ensures blood continually meets water with a higher oxygen concentration. Number three, the concentration gradient is maintained. So think back to Ficks Law, meaning that oxygen continues to diffuse from the water to the blood, and there's that really useful little phrase, along the whole length of the lamellae. Okay, I'll just leave that there for a second. If you can learn those 3 sentences verbatim, you can't really go wrong with countercurrent exchange. Okay, so here's some different types of gill. This is an internal one, an external one. So we've been looking at internal up to now. That's an axolotl, isn't it? They've got external gills. Again, though, we can see lots of branching with those filaments, lots of lamellae on those filaments to increase the surface area. Okay. Lastly, just describe and explain how gills are adapted for efficient gas exchange. Typical type of question that you might get. And we've mentioned all of these things already. Okay. Is that everything I wanted to do? Yeah. Right. Thanks ever so much. If you've got any questions, give me an email or right below. Hope everything's going well. Cheers.